Assalamualaikum and greetings to my lecturer, Dr. Anwar Muhammad Zain. My name is Amni binti Hari Anwar and I'll be presenting about my e-portfolio task for this whole semester. For the first week, we learned about the hematopoietic system. So we learned that blood is a specialized connective tissue composed of cellular elements suspended in a liquid extracellular matrix called plasma. So plasma, which is 90 to 92% of water, which acts as a highway for transporting nutrients, hormones, gases, and metabolic wastes like urea. So the cellular components includes the erythrocytes. So these biconcave anucleated discs have a shape that optimizes the surface area for gas exchange and allows them to fold when moving through narrow capillaries. So each red blood cell contains of millions of hemoglobin molecules which reversibly binds oxygen and transports it to lungs, to tissues. So next is the leukocytes. So these includes phagocytes like neutrophils, which digest bacteria, and lymphocytes, which produces antibodies to neutralize pathogens. So lastly, the platelets, also known as thrombocytes. So these are fragments of megakaryocytes that form platelet plugs to seal injured vessels and releases procoagulants to activate the coagulation cascade. So for the next week, week 4, which is blood grouping, we learned that blood grouping is the classification of blood based on the presence or absence of inherited antigens on the surface of the red blood cells. So the ABO system categorizes blood into 4 groups: A, B, AB, and O. So while the RH factor identifies the presence or absence of the D antigen. So transfusion compatibility is vital. For example, the Rh negative individual should only receive Rh negative blood to prevent immune reactions. If incompatible if incompatible blood is transfused, the recipient's antibodies triggers agglutination, which is clumping, and hemolysis. So this reaction can block blood vessels and causes fatal damage to the kidneys. So the first practical of this semester is practical 1. So I am in group 2A3. So practical 1, which is the cardiovascular anatomy and the cardiac measurements. So the heart functions as a 4-chambered pump where deoxygenated blood flows from the vena cava to the right atrium and ventricle, which oxygenated blood is ejected from the left ventricle into the aorta. So in clinical measurements, we calculated the basal metabolic rate, the BMR, to estimate resting energy needs, and the mean arterial pressure, MAP, to ensure adequate tissue perfusion. The cardiac output, CO, was determined by multiplying the heart rate by stroke volume. So results showed that our cohort maintained a normal MAP range from 78 to 95 mmHg. So through an exercise optimization task, we found that a pulse recovery drop of more than 21 beats per minute indicated as good cardiovascular status, whereas a drop of less than 12 indicated as a risk. So the next practical, we did this at the dissection lab at the medical campus. So practical 2, in this student activity pack, we learned that the respiratory system facilitates gas exchange at the alveoli, where oxygen diffuses into the blood and carbon dioxide is exhaled. So this mechanism really relies on the diaphragm contracting and moving downward during inhalation to increase chest volume and decreases internal pressure, allowing air to flow in. So case studies illustrates the breathing difficulties often arise from lung inflammation or airway blockage. Additionally, poor circulation can lead to edema, which is swelling when the heart fails to pump efficiently, causing fluid buildup in the tissues. So the next practical is the cadaver viewing. So in the dissection lab, a direct observation of the cadaver confirmed that the heart is a firm muscular organ approximately the size of a palm. So we trace the positions of the valves, including the tricuspid, bicuspid, pulmonary, and aortic, which ensures one-way flow. So the next ePortfolio task is week 8, which is the histology of the GIT layers. So the gastrointestinal tract GIT consists of 4 universal layers, or also known as tunica. The tunica mucosa features an epithelial transition from stratified squamous epithelium in the esophagus for abrasion protection to simple columnar epithelium in the stomach and intestines for absorption and secretion. The muscularis mucosae contains the pacesetter cells that triggers rhythmic contractions to increase surface area for nutrient absorption. The tunica submucosa contains the Meissner's plexus, which acts as a local control center for glandular secretions and blood flow. So the next ePortfolio task is week 9, the digestive system. So the digestive process transforms a bolus into chyme through mechanical and chemical breakdown. In the oral cavity, mastication and salivary amylase initiates the process. So the bolus is moved by peristalsis down the esophagus to the stomach, where pepsin and hydrochloric acid begins the protein digestion. So mixing waves occurs every 15 to 20 seconds to macerate the food. In the duodenum, chyme mixes the bile for fat emulsification and pancreatic juice to resume the carbohydrate and protein breakdown. The jejunum and ileum utilizes the brush border enzymes and villi to complete the nutrient absorption. So the next practical is the digestive system stations, where the following alimentary canal we identified the small intestine as a 6-meter long tube with specialized circular folds and villi for maximum absorption, contrasting with the large intestine, which focuses on water and electrolyte absorption. The liver is a vital accessory organ that produces bile and deoxifies blood and stores glycogen. The gallbladder concentrates and stores the bile until it is needed in the duodenum. The pancreas functions as a mixed gland as Cynar cells secretes digestive enzymes, which, while the islets of Langerhans contains beta cells that produces insulin to regulate blood sugar. So the last week ePortfolio task is week 10, the urinary system. The nephron is the functional unit of the kidney where the filtration occurs in the renal corpuscle. The RAAS system is a critical regulatory mechanism when the juxtaglomerular cells detects low blood pressure, they release renin, which triggers a cascade leading to angiotensin II production and the aldosterone secretion. Aldosterone causes the kidneys to absorb sodium and water, increasing blood volume and restoring pressure. Histological study of the bladder reveals the transitional epithelium with umbrella cell that flattens to accommodate up to 500 ml of urine. So lastly, the practical 4 is the urinary system stations. So internally, the kidney is organized into cortex, medulla, and renal pelvis. Within the nephron, the proximal convoluted tubule (PCT) is the main site of reabsorption, lined with the simple cuboidal cells featuring numerous microvilli. Urine is transported via the ureters through rhythmic peristaltic contractions of the smooth muscle. The urinary bladder causes the detrusor muscle to expel urine through micturition, while trigone region directs the flow towards the urethra. Clinically, the female urethra is shorter than the male urethra, which contributes a higher risk of urinary tract infections in women. Thank you. That is all from me.